Hi biologists, let's start by looking at the learning outcomes for this lesson. At the end of this lesson and following the biology syllabus, you should be able to describe the formation of endospores. What does this actually mean? What are we trying to learn? Well, basically, it's very straightforward. All we have to be able to do here is to be able to describe how endospores are made. Let's talk about endospore formation. Before we discuss endospore formation, we might need to go back and revise bacterial reproduction. If you recall from a previous video, bacteria, unicellular organisms, being only one cell big, reproduce asexually by binary fission. Asexual reproduction means that there is only one parent involved and the cell basically splits in two by mitosis or binary fission. This process of reproduction or binary fission occurs every 20 minutes in optimum conditions, when the bacteria have plenty of food, when there is a suitable temperature, when there's oxygen and when there's moisture. Don't forget, the resulting daughter cells are identical to each other and to the original cell. If you like, the resulting daughter cells or new cells, daughter doesn't have anything to do with female, it's just a science word for new cells. So the resulting new cells are identical to each other. Therefore, they're called clones. You might like to refer to a previous video if you would like to revise this section. Basically, this is how bacteria normally reproduce. They reproduce by binary fission. However, when conditions become harsh and less favourable, perhaps there's a lack of water or drought, maybe there's a lack of food, maybe it's too hot or too cold, maybe the conditions are freezing, or they're subjected to radiation. In other words, they are exposed to adverse conditions. You need to know examples of adverse conditions or unfavourable environments. In that case, some bacteria are capable of forming endospores. Now, the important thing to note here is that technically speaking, endospore formation is not reproduction. It really is a survival strategy as such, undergone when conditions are harsh. Now, endospores are resistant structures with a very low metabolism, meaning that endospores are difficult to kill and they keep ticking over, barely existing if you like, because their chemical reactions go on at such a low or slow rate. Therefore, they can survive a very long time. They can survive for hundreds of years in actual fact. Now, when conditions become suitable again, when the temperature is suitable, when there's plenty of oxygen, there's moisture or water, and there are plenty of nutrients, or the bacteria have plenty of food, then the endospores will germinate when the conditions become favourable again, and we will create new bacterial cells. So let's have a look at the process of endospore formation. We can see here a typical rod-shaped, shape of a tic-tac, a rod-shaped bacteria with the bacterial cell wall. Now, when conditions are harsh, the contents of the bacterial cell will shrink and round up and form a structure within the original bacterial cell. Prior to this, the DNA would have replicated, but we don't necessarily need that detail once we understand that in unfavourable conditions the bacteria stop reproducing normally and will form an endospore by the contents shrinking and rounding up. The original cell, which is no longer needed, the original cell wall, which is no longer needed, basically disintegrates away and the endospore is set free. When conditions are favourable, this endospore will then swell up with water, burst the walls and reproduce in the normal fashion by binary fission. So let's have a look at a step-by-step description of endospore formation, accompanied with a labelled diagram. As we've already said, bacteria will form endospores when they are experiencing a harsh environment or adverse conditions. In such unfavourable conditions, the bacteria stop reproducing. The contents of the cell will shrink, they will round up, forming a thick wall within the original structure. This structure is the endospore. The original cell wall disintegrates and the endospore will then be released. This endospore, as we have said, can survive for a very long time, and in favourable conditions the endospores will absorb water, break their walls and then reproduce by splitting in two, forming two new bacterial cells by binary fission. As an added extra, for those of you able for a bit more, what's rather interesting is that some pathogens can make endospores. This makes it very difficult to eliminate such pathogenic or disease-causing bacteria. For example, Clostridium botulinum causes botulism or food poisoning. Endospores are not actually killed at 100 degrees Celsius, which means they're not actually killed by boiling. An autoclave has to be used to destroy endospores. An autoclave is a machine that uses steam under pressure to kill pathogenic or disease-causing bacteria, fungi and viruses. Autoclaving at 121 degrees Celsius for 20 minutes is actually necessary to destroy endospores. Autoclaving is often used to sterilize lab and hospital glassware and equipment. And there you have it. Practice in a jotter. Remember, when the going gets tough, the tough get going. Now that we have reached the end of our lesson, have we achieved our objective? Can you describe the formation of endospores?